ሄሎ እንዴት ናችሁ? ሰላም ናችሁ? እንግዲህ አሁንም ቫይሮሎጂ ላይ ነው ያለነው። ቫይሮሎጂ እንግዲህ ከዚህ በፊት ምን አየን? ጀነራል ኮንሰፕት ኦን ቫይረስስ ዋት አር ቫይረስስ የሚለውን ፈርስት ሌክቸር፣ ሰከንድ ሌክቸር ደግሞ ቫይራል ሲሜትሪ። ዛሬ ደግሞ ክላሲፊኬሽን አላቸው ቫይረሶች። ክላሲፊኬሽን ዴይ ዱ ሃቭ ክላሲፊኬሽን። የቫይረስ ክላሲፊኬሽን ያው ቫይረሶች ሊቪንግ ኦርጋኒዝም ከሴል አይሰሩም፣ ሊቪንግ ኦርጋኒዝም አይደሉም። ጀስት የራሳቸው የሆነ ክላሲፊኬሽን ስኪም አላቸው እና አንዱ ነው ማሳያችሁ ዌል ናውን የሆነውን ዲኤንኤ ቤስድ ኦር ኒውክሊክ አሲድ ቤስድ ክላሲፊኬሽን የባልቲሞር ክላሲፊኬሽን ነው። እንግዲህ ወደ ሌክቸሩ ከመሄዳችን በፊት እንግዲህ አዲስ የሆናችሁ ይህን ገጽ ሰብስክራይብ አድርጉ። ደንበኞች የሆናችሁ እንግዲህ ላይክ፣ ሼር በማድረግ ተቀላቀሉን። ጥያቄ ካላችሁ ከስር ጥያቄ መጠየቅ ትችላላችሁ። ኮሜንት ማስቀመጥ ትችላላችሁ። ለጥያቄያችሁ መልስ ሰጣለን። ለኮሜንታችሁ ሁሉ ቀድመን እናመሰግናለን። ቴንክ ዩ ቬሪ ማች። ወደ ትምህርታችን ሂድ እስቲ። ሌትስ ጎ ቱ አወር ሌክቸር and therefore as I have told you today classification of viruses ነው የምናወራው። ክላሲፊኬሽን የትኛው ክላሲፊኬሽን? ኦፍ ኮርስ when we talk about viral symmetry አንዱ ራሱን የቻለ በሲሜትሪ ክላሲፊኬሽን ነው። we classify viruses based on the symmetry of their capsid coat into three categories እንዳትረሱ። that is polygonal፣ ይሄ ፖሊሄድራል ስፔሲፊካሊ አይኮሴድራል እና ሄሊካል ፕላስ ኮምፕሌክስ ሲሜትሪ ቢባሉ እና ይሄኛው ክላሲፊኬሽን ኦፍ ቫይረስስ ቤዝድ ኦን ዴር ጄኔቲክ ማቴሪያል ስፔሻሊ የማንን ነው? የባልቲሞር ክላሲፊኬሽን ስኪምን ነው እና ባልቲሞር ክላሲፊኬሽን ኦፍ ቫይረስስ ይባላል። ሌትስ ሴይ ሊትል አባውት ባልቲሞር ክላሲፊኬሽን. Baltimore classification is a system used to classify viruses based on their manner of messenger RNA synthesis. Based on their manner of messenger RNA, mRNA synthesis. If you are asked what is the basis for Baltimore's viral classifications, it's based on manner of messenger RNA synthesis ነው። and this approach to virus classification was first suggested by David Baltimore in 1970, after whom the scheme is named. This classification scheme is named after this personality, David Baltimore, the American virologist, genetist. And let us little talk about David Baltimore. Who is David Baltimore? This is the personality. This is the photograph showing this man, of course, still alive. He is still alive, though he is retired, he is still working. And David Baltimore is an American biologist and 1975 Nobel laureate in physiology and medicine. He won the Nobel Prize in Physiology and Medicine in 1975. Of course, Baltimore won the Nobel Prize with Renato Dulbezo and Howard W. Tamin for the discovery of the enzyme. This enzyme. These three shared the Nobel Prize for 1975 for discovering reverse transcriptase. Do you remember this enzyme? This enzyme is that vital enzyme for retroviruses like HIV. This enzyme is able to make DNA from RNA, single-strand RNA. You see, we have talked about retroviruses. Retroviruses are positive sense single-stranded RNA viruses. When they get into the host cell, their reverse transcriptase enzyme can make double-stranded DNA from their single-stranded positive sense RNA. And this viral DNA, viral enzyme was discovered by these three personalities who shared the Nobel Prize for medicine in 1975. Notably, it says, reverse transcriptase is an enzyme that converts RNA into DNA, playing a crucial role in the life cycle of retroviruses and various molecular biology application. Still, we are using, it has got, the enzyme has got various molecular biological applications. And let us see the classification. Classifying viruses by how they relate to messenger RNA. You see? How they relate to messenger RNA. You can see here. DNA viruses, class 1, class 1 double-stranded DNA virus. This makes just transcribes to produce messenger RNA by using the sense strand, of course. And the second group are single-stranded RNA, I mean DNA viruses. Single-stranded DNA viruses first make double-stranded, first make their single-strand DNA into double-stranded DNA, then they make messenger RNA. The others are RNA viruses. RNA virus. The third group, the RNA virus, are double-stranded RNA viruses, which use one strand as messenger RNA. The other, class 4, RNA viruses are the positive sense, I think, the positive sense RNA viruses, which are directly used as messenger RNA for protein synthesis. Classifying viruses by how they relate to messenger RNA, of course. Then here you can see the first two classes, class 1, class 2. Class 1 is double-stranded DNA. You can see the positive sense and the antisense, just like our genome transcription. It makes messenger RNA and directly produces protein. The second, single-stranded DNA. The DNA could be the sense strand or antisense strand. Anyway, whether it is sense and antisense, it makes its DNA first double and produces messenger RNA. The third group are RNA viruses, which are double-strand RNA viruses. They use one strand, the coding strand, the one strand as messenger RNA. The fourth category are positive sense RNA viruses, which which use negative sense to make positive sense, and the positive sense directly is used to protein synthesis as messenger RNA for protein synthesis. The fifth is negative sense. The negative sense is directly used. The negative sense is directly used to produce messenger RNA. Messenger RNA, so that the messenger RNA is used to produce the protein of the virus. And the last is positive sense RNA, which makes negative sense, and from these two makes DNA, double-stranded DNA. The retrovirus groups are, according to Baltimore's classification, the retroviral groups are the group 6. They have plus sense RNA, single-strand RNA. This makes its partner negative sense, and from these two, DNA strands are made, and the two strands of DNA made on this RNA are used as genetic material of the virus, of course. This is Baltimore's classification. 7 Baltimore groups are described that make into consideration, it says, whether the viral genome is made of deoxyribonucleic acid DNA or the ribonucleic acid RNA, and whether the genome is single or double-stranded. You see, in this classification, these criteria are used, and whether the sense of a single-stranded RNA genome is positive or negative. These are some double-stranded DNA virus, single-stranded DNA virus, double-stranded RNA virus, single-stranded RNA plus virus, the positive sense, single-stranded RNA minus negative sense, and single-stranded RNA plus RT viruses, plus RT, positive says, plus reverse transcriptase. Those viruses which are single-stranded RNA positive cells, but when they enter into the cell, they convert their RNA molecules into DNA. RT stands for reverse transcriptase, the enzyme. And last, double-stranded DNA, but gapped, which are circular. The top one, the first one is double-stranded DNA linear. This is circular, but it has a gap, a gap which is not double-stranded. The double-stranded DNA gapped. There are some small sections in the circular DNA where the DNA remains single-stranded or not double-stranded. You can see some examples: double-stranded DNA viruses, pox viridae, herpes virus, papilloma viruses. These are associated with cancer, cancer of the reproductive systems, especially females. Polyoma viridae associated with cancer also. Adenoviridae associated with cancer also. These are all carcinogenic viruses, cancer-causing viruses. The second, single-stranded DNA viruses, some examples: adeno-associated virus such as circoviridae, ወይንም circoviruses, parvoviridae, parvoviruses, very, very small. These viruses are very, very small and depend on the presence of adenoviruses to multiply. Usually infect insect. You see, these viruses go into host cells where adenoviruses are present. They need the help of those adenoviruses for their complete life cycle. That's why we call them adeno-associated viruses. You see? Otherwise, if adenoviruses are not found together with these single-stranded DNA viruses known as viruses and parvoviruses, they can't multiply and complete their life cycle. That's why they need a friend together with those, a friend. These friends are adenoviruses. The third group, double-stranded RNA viruses. These are examples: Reoviridae. These are enveloped, enteric, or respiratory. They can affect the GI tract or the respiratory tract. Could be enteric or respiratory. Example, rotavirus. Rotavirus is one well-known GI tract diarrhea causing especially lethal to little children, watery diarrhea, GI tract infection. Single-stranded RNA plus sense or positive sense viruses. These are many very important group as pathogens. This group is really smart, contains smart group, smart in a sense they are effective in causing disease. Like coronavirus, the SARS-CoV-19, the SARS stands for serious acute respiratory syndrome. Of course, coronavirus belongs to this group. Flaviviridae, like hepatitis C virus. Picornaviridae, example, polioviruses, hepatitis A virus. Rhinoviruses, common cold. Aftovirus, foot and mouth disease in cattle. The aftovirus is the common infecting virus, cattle infecting virus, which causes foot and mouth disease in cattle. Wound, it causes infects the foot area and the mouth, causing wound inflammation. Rhinovirus is also here, common cold virus. Single-stranded RNA viruses. Then single-stranded RNA negative cells. These are orthomyxoviridae, like influenza virus is single-stranded RNA negative cells. Paramyxoviridae, measles in children. Rhabdoviridae, rhabdoviridae are those which cause this rabies, the hydrophobia or rabies. Flavoviridae, Ebola virus is in this group. Flavoviridae, the Ebola virus is the hemorrhagic virus. Single-stranded RNA plus RT. These are the retroviruses. The retroviruses, viridae retroviruses, and these are the group which have positive sense RNA, single-stranded, but change into DNA, double-stranded DNA when they enter to the host cell. Okay, the host cell, and the last double-stranded DNA gap, circular double-stranded DNA viruses are have a gap in one strand, which is repaired to create a complete double-stranded DNA genome prior to transcription. When they before they begin transcription, the gap is sealed, otherwise it's gapped. Hepatadenoviruses, example hepatitis B virus belongs to this group. Hepatitis B virus, please. Except hepatitis B virus, the other hepatitis viruses are RNA viruses. Hepatitis B virus, which is transmitted by sexual contact, usually, and lethal, of course, it has vaccination, though it is expensive vaccination, it has vaccination. It's lethal, killer. These are the 7 categories of Baltimore's viruses, or Baltimore's classification of viruses based on their genetic material, specifically based on how they are related to the messenger RNA. And families in general of virus infecting vertebrates, you can have even you can classify viruses based on their host, human viruses, insect viruses, plant viruses, bacteria viruses. Therefore, they are highly host specific. Therefore, there are a group of viruses which attack vertebrates, which attack invertebrates, but specifically some insects, and even some species of plants, some variety of plants. These are some description for your extra knowledge. If you are interested, you can look at them, appreciate, and even it may inspire you to join virology if you are interested in the study of the pursuit of the knowledge of biology, of course, the future. Virology is really, there is vast area for research if you join the virology department of biology, of course. Okay, thank you very much. Here we wind up our third presentation on virology. And next, I will come to the same lecture on virology on another topic. Thank you very much.